So François Renard is a leading geoscientist, internationally recognized for his work on rock deformation, fracture processes, and advanced imaging on geological materials. He's professor at the University of Oslo, affiliated with the Universitetet Grenoble-Alpes in France, and director of the NJORD Center, which is an interdisciplinary research center focused on the physics of geological processes. François has been a pioneer in applying time-lapse and multimodal 3D imaging, what we now refer to as 4D imaging, to better understand geological processes as they happen. Today, he will share insights into recent advances in 4D imaging, as well as the challenges associated with data acquisition and processing. So, François, please take the floor.
Thank you, Céline, for this nice presentation. So I'm just sharing my screen. Can you see? Céline, can you see now?
I hope so. I can't see your screen. Now I see it happening. Yes, now I see it.
Excellent.
It's working.
So thank you for the invitation and for allowing me to present this field, which is about, so today we will discuss about 4D imaging, and I put a movie on the top right side to show what we will discuss about. So it's a rock sample. Here it's carbonate under condition of pressure and temperature at depth. And that is imaged through time in 3D. And that's what we call 4D imaging. And what we see on the left side, it's the outside of the sample with the piston on top and bottom. And on the right side, that's the interior of the sample with, when deformation occurs, an increasing number of microfractures that will accommodate the growth of a fault. And this fault grows slowly. It's a bit like a slow earthquake. But more generally, my presentation today is part of a big change that occurred in earth sciences in the past 20 years, which has been the data revolution. Data revolution because now we observe the Earth with so many different tools, satellite, GPS, oceanic bottom seismometer, drones, seismology network, and so on. And part of these tools is these large international facilities, cyclotron, neutrons, and these tools allow to reproduce conditions of pressure, temperature, fluid chemistry at depth. And it's now possible to see inside sample what happens, which was not possible 20 years ago. So this talk is about using user facilities. And user facilities, there are many in Europe. There are many opportunities for people to do experiments. I just listed three. These three, they have a property in common, is that several facilities are on the same site. So there is one in Switzerland, the Porous Institute, with 3 facilities: an X-ray source, a cyclotron, a neutron source, and a X-3 electron laser, a laser, high powerful laser. There is another one in Lund in Sweden with a cyclotron and the neutron source that is now open to users. And the third one in Grenoble with the cyclotron and the neutron source. In Europe, there are around 10 cyclotrons, 10 neutron source, and 3, a bit more, X-FEL facilities. And all these facilities, they function the same way as Excite. People apply a couple of times per year, send a proposal, then there is a selection. And when the proposal is granted, the beam time is given for free to the users. The talk today will be about X-rays and neutrons. And perhaps to start, just what's the main difference between the interaction between these X-rays and neutrons with matter. X-rays interact mainly with the electrons, so the electronic field around an atom. And this X-ray, they can be either absorbed or scattered. And if they are sent on the left and collected on the right, the absorption, for example, is given on the periodic table by the color. Dark means it's absorbed a lot, and white, it means it's absorbed very little. Neutrons, on the opposite, they interact with the nucleus of atoms, with the center. They don't interact with electron. A neutron is not charged. It doesn't interact with the positive or negative particles in the atoms. And again, they can be either absorbed or scattered. And that's the same periodic table with the same color code, where dark means lots of absorption and white, very little absorption. And perhaps what you can see, right, for an element like hydrogen, hydrogen is very little absorbed by X-rays, so X-ray will not see it, whereas it's absorbed by neutrons, so neutron will see it. And if we take a rock, a silicate, silica is absorbed by X-rays, but is very little absorbed by neutron. So combining these two techniques, X-ray and neutron, allows to image different elements because they have different absorption properties with respect to this kind of X-rays and neutron. Neutron and X-ray, you can be used to acquire radiograph, like what we do in the hospital. And that's a radiograph, a 2D view of the same object. It's a glass jar with inside a little plastic figure, a Playmobil. And this Playmobil is surrounded by staples. The neutron, they see very well the hydrogen, so the plastic of the Playmobil. They don't see much the staple and they almost don't see the glass. The X-ray, they see a bit the glass, they see the staples, but they don't see the Playmobil. So using these two imaging techniques at the same time to image the same object, we can get different information. And I will come back later to this example. It's a porous basalt. So it's a core. All the dark spots is the porosity, it's vesicles in the basalt. And this basalt, when imaged with tomography using neutron, we can see the light color is some water present in the pore. The dark color is pores that are empty, and the dark gray is the solid. So the empty pores and the solid, we almost don't see them, but we see very well the fluid. And if we use X-ray imaging, it's the opposite. We see very well the solid, the matrix, and what is in the pores is more difficult to see. So what has been this innovation, which is called 4D imaging or 4D in situ imaging, is the capacity to follow processes in 4D. So that's a core sample of sandstone that is loaded in a special triaxial apparatus until the sample will fail. The diameter is 5 millimeters. What appears in white is the grain of quartz in the sandstone, and blue, that's the porosity. And perhaps you see that the failure in this system occurs by the propagation of small fractures until they coalesce to form a fault. And when this fault is formed, the entire sample can collapse. So how do we do that? We need first an apparatus. So that's the latest we have built at the European Cyclotron with Benoît Cordonnier taking care and building this apparatus. So the sample is inside this little part. And there are some pumps below, electronics below and above that can apply confining pressure. So here we did a test where we reached 872 megapascal of axial pressure for a sample at 90 MPa confining pressure. That was a granite, and I think it broke at this condition. Everything is mounted on a rotating table instead of on the beamline of the cyclotron, because to do X-ray tomography, the idea is to rotate the sample, acquire thousands of radiographs, and reconstruct the sample in 3D. And to do this acquisition as a function of time, so to get a number of 3D images, typically in an experiment like that, we will get between 50 to 100 scans to do the experiment. There is one challenge, a bit of challenge, is that doing this, we are buried with data. 3 days of experiment on this machine is 100 terabytes of data. So we need both to define a strategy to not be buried. And that's why all the students that have been working, or early career researcher in my group, they have acquired the data scientist competencies to handle this data and to reduce this data, to transform this data into an information that is useful to solve scientific questions. And the rest of my talk now will be about 2 scientific questions we have solved in the recent years. One of them is what happens before an earthquake. It's called the preparation process. Not only before an earthquake, but before a geomaterial breaks. And what happens is that some microfracture, some damage will accumulate. And when sufficient damage has accumulated, the fracture will propagate. So we did that on several kinds of samples. For example, we use this sample, which is a cylinder with 2 notches, which would represent what happens at the tip between 2 propagating faults before they link. There is a rock bridge, and this rock bridge will damage slowly until it breaks. So we did an experiment, Irina Prastiani did this experiment, where the sample, it's a 1 by 2 centimeter piece of granite between 2 pistons with a confining pressure. The notches are on the 2 sides of the sample. And what appears in blue in the 4 different scans at the bottom is this microfracture that develop until the sample break. And with these X-ray images, it's possible to extract each of these fractures. On the left, that's the stress strain curve, so that's how much stress is put on the sample. The sample is shortening by something like 1%. So there is a shortening of 1% before this rock bridge will break by the coalescence of all these microfractures. So in 3D, that's the last scan before failure. So outside the granite sample, some microfractures can be seen. And inside, all these microfractures, they are almost vertical. They align with the vertical loading, but they will form this fault zone that will connect the 2 notches and will break this rock bridge. What can be done also with this technique is to follow individual fractures, and that's the work that Jessica did. It's just published this month. So take an intact rock sample, that's also a granite here, and a core sample, which is loaded until failure. It's possible to see all the cracks. So the different grayscale here is different minerals. This one is an oxide. This one is a biotite, and the background is a mixture of quartz and feldspar. And it's possible to see all these fractures and to follow the fracture that nucleate, the fracture that propagate, and the fracture that coalesce. And doing this, it's possible to identify which fracture will be responsible for the final failure of the sample. And in this case, the final failure is a coalescence of a few fractures that will coalesce and that will lead to a failure at the system size. If we now go deeper in the Earth's crust, there is this transition where the rock goes from fully brittle to semi-brittle to ductile. It was described in the early 70s, late 60s, early 70s by the group at the USGS and at MIT. And this transition is the transition that occurs above and below the seismogenic zone. At some depths in the lithosphere, there is no more earthquake. And one of the reasons is that there is a transition from the brittle properties of rock to fully ductile. So that's 3 samples of Carrara marble. They form at different confining pressure, so A, B, and C. And they have been scanned after the experiment. The size of this sample is 4 by 8 centimeters. So that's a static view of the deformed sample. But experiments now allow to do these experiments in situ. So that's the result of 3 experiments. 3 experiments of Carrara marble. The one on the top is in the brittle regime. So in the brittle regime, I don't know why, but the movie doesn't play, but let's continue like that. In the brittle regime, the differential stress is increasing, and the sample will accumulate deformation until the sample break. In the semi-brittle regime, the stress will increase and then the sample will start to flow. So in this case, the sample has been shortened by 10%, which is quite a lot for a rock, to shorten a solid rock by 10%. And this one, at 70 MPa confinement, the sample keeps also deforming. And during this deformation, the sample can be followed. And what was interesting in this study that has been published this month is that the sample accumulates a lot of porosity. So the way to deform the sample here is to create a sample with 18% porosity, a bit like a sponge. And when the sponge is deformed, it's much softer, so it's easy to deform. Let's move now to the last topic, which is how to follow fluids in a rock. So we have seen that with X-rays, we can follow the solid. And with fluids, we can follow with neutrons, we can follow how fluid circulates. And the motivation of this study was in the context of mineralizing CO2 in the form of carbonate. What happens if CO2 and water is injected in a basalt like that? And with time, the CO2 will react with the magnesium, calcium, and iron of the basalt and mineralize that basalt. So this one is a natural sample where all the part of the vesicle, so what is black is the porosity, but part of this porosity has been filled with calcite. And this sample, they are seen offshore Norway, but they are seen also in the field. And that's the Permian basalt from the Oslo fjord, where all vesicles, or almost all of them, have been filled with carbonate. And the question is, what are the conditions of fluid circulations to do this mineralization? And the way we do that is we develop a set of experiments where we put a core sample of basalt, this one is 5 by 5 centimeter, perhaps 30% porosity, inside a core holder here at a room temperature with almost no confining pressure, but fluid injection. And on one inlet, we inject fluid with calcium, and the other inlet, we inject fluid with carbonate. And when the two fluids, calcium and carbonate mix, they will precipitate calcite in the sample. And there is some outlet here to collect. And what we do 3D imaging through time of the fluid inside the sample. And the question we asked was, how do these two fluids mix inside the sample? And when they mix, can we see the mixing interface? And with neutron, it's possible to do that. So that's the basalt sample. We injected two fluids. One fluid is heavy water. So heavy water, it's water where the hydrogen has been replaced by deuterium. So deuterium is hydrogen with an additional neutron. So that's the blue fluid. The green fluid is a mixture of normal water and heavy water. And the result of that is possible to see the two fluids in the porous medium. And what we expected was that at the interface of the two fluids, calcite would precipitate. So for this experiment, there has been a lot of challenge to make it happen. But one way to image the outcome was to take the sample after the experiment and to put it under a cyclotron. So it was possible because the experiment was made in Grenoble on the neutron source and the sample was moved to the cyclotron. This sample was scanned in 3D. So the black is the porosity, the blue is the precipitate, and the white is the solid. And if we zoom in, for example, we can see some pores without precipitate, but this pore is filled with calcite. And in the movie below, what we see is a fusion of the neutron images and the X-ray images. So we could put the two scans, the neutron scan and the X-ray scan, into the same referential. So with the X-rays, it's possible to see the solid. With the neutron, it's possible to see the fluid, where the fluid is going to circulate inside the sample. And after 6 months, to image where the precipitate occurs inside the porous medium. As I mentioned you, there is a challenge with that data. Just this volume here is 4 terabytes. So it means that a normal computer does not have the memory even to load the entire volume. So with this, I will stop now. I would like to thank all the team. What I have shown you has been the work of a team for the next 5 or 6 years. So the team was in Oslo a couple of years ago, and we could make this picture together. This team is distributed in many places in the world. And if you're interested to read more, I put also some publications we did last year and this year, and that show the results of what I have presented today. I thank you for your attention. I am ready for questions.
Fantastic, François, and very nice imaging there of a precipitation of calcite. It's very interesting. So just really interested to know, do you really have then your sample for 6 months in the machine to be able to control all the variables, or is it kind of like put it in a cupboard, or and then bring it back to do measurements? How do you
So
do
no,
that in practice?
so we did the experiment, and when you do an experiment on a neutron source, the sample is radioactive. It becomes radioactive, so you cannot touch it for some time.
Right.
So we had to wait for the radioactivity to decay, so we just let it in the neutron source for 6 months, and after 6 months it was less radioactive, so we could bring it to the X-ray source. That's how it happened for this sample.
Ah, okay. Yeah. So you don't have X-ray maybe of the kind of like of that first time point. You only have X-ray measurements of the later time point.
So it's no, it's a bit different. We have X-rays. I don't know if you see that. Can you see the PowerPoint?
Yeah.
So on the neutron source, there is both the neutron and there is a laboratory X-ray scanner. So it's possible to get an X-ray acquisition. The issue is that usually these sources, they are less powerful than a cyclotron. So the quality of the image is not sufficient to detect the precipitates in that case, because our sample is quite big. So we have to make a compromise when we use this dual system on neutron source. And usually we favor the neutron because it's quite exceptional to be able to see the fluid.
Nice. Yeah. Yes. So also another question here is, so how do the laboratory observations of this damage production that you showed, so that happens, I think, during earthquakes. So how does that translate into the natural kind of environment? Is it really, can you really in the laboratory environment recreate these kind of natural occurrences?
So I think what we see in the lab, it's partly what happens in nature in the weeks, months, years before an earthquake. Is that there is a zone inside a fault, it could be a landslide or a glacier as well. There is a zone that will weaken slowly. And the weakening process under this condition is to create microfractures. So I think we reproduce what should happen at depth. It's very difficult to see it at depth. One of the reasons is that all these microfractures, most of them, they are silent. So seismometers will not see them. And that's something that is well known in laboratory experiments. Colleagues would do experiments by adding microphone or little seismometers in the lab. What they record with seismometer is between 1% and 8% of the total strain. So it means that more than 90% of the strain is silent.
Wow.
So that's why seeing them in the experiment is quite an added value to understand the processes.
Fantastic. So what are the biggest challenges in ensuring data consistency over long experimental timescales? Because you already mentioned the terabyte scale of course, getting the data. So is that the biggest challenge, or are there more? That is just simply not, well, it will just create a lot of data in itself, or are there more challenges?
It's not the challenge of the data is usually the amount of data. So it's to be prepared when we do these experiments to work on data that are very big. Neutron data, they are not that big. Usually we take a couple of hard drives and then we can take them back. The cyclotron data we have now are too big to even transfer them. So we start to work directly on the data on the cyclotron and to do a data reduction. So to go from 100 terabyte to 10 terabyte, for example, to automatize this workflow such that data can be handled. On the consistency of the data, the way we do that is that every time we publish a paper, we publish the data.
Great.
So that's a way to, we create a repository where people can access the data. And then the consistency of data, it's we don't control it too much because the data format is evolving with time. So we just adapt ourselves to this evolution.
Okay. So I saw that there's a question in the chat as well. It's regarding carbonate precipitation experiments in basalt. Will this kind of carbonate precipitation from fluid-fluid mixing be completely different from CO2 brine basalt reaction in terms of distribution of precipitation and morphology, etc.?
That's a very good question, and that's I think it's an ongoing topic. It's for me, I still think that this mixing process in a real CO2 operation is relevant, because what happens, it's what is injected is at least in CarbFix, for example, it's a carbonated water, so it's water rich in CO2, and this water will meet fluids in the basalt that have a different pH, pH 10. They start at pH 4 and then mix with pH 10. And probably that's at the interface between these fluids of different pH that mineral precipitation will occur, will be more favorable. So I still think there is a fluid mixing process. It's just that what I show you in this movie, we cannot see it at depth with this resolution. So that's why it's important to do laboratory experiments.
Definitely. So this is this kind of like pH difference. Is that also something that they use in this technique as well when they inject CO2? Use the pH difference to
I
don't know, inject.
For CarbFix, they did it the other way. They tried and it worked. So now it's a question of understanding why it works.
Okay. So now you can actually recreate these kind of conditions in a laboratory setting.
Exactly.
Fantastic. So I hope that answers your question. Please, if there are any more questions, let me know either in the chat or in the Q&A, or raise your hand. I see another question. So 4D means 3D constructed model over time. What if some activity happens in a very short time, for example, reputation? Technically, is it possible to capture the time-lapse 3D model, microseconds, probably microsecond scale? Does that make sense?
Yeah. So
okay.
When you do this experiment, you have to design how you load a sample, so the rate at which you load, that is compatible with the rate it takes to get a 3D scan. So this kind of 3D scans here, to give you the entire experiment, each of them lasts for between 8 and 10 hours. We have perhaps 100 scans acquired, and each scan is 3 minutes. And I just show here only a few of them because we don't use all the scans. It's possible to do ultra-fast imaging. And we did that, for example, when you send a shock wave on rocks, which could mimic what happens at the tip of an earthquake or the impact of a meteorite. And for this, there is a technique which is called ultra-fast imaging. And now the record we have is to get 4 million images per second. We will soon be able to go to 15, but only in 2D. So taking a 2D radiograph of a sample more or less when it explodes. So it's possible. What is possible also is for very specific samples to do a scan of a small course in 3D, so a bit like this one, with a few seconds, a scan in a few seconds. But it has to be a kind of small sample. And so there are techniques to accelerate time. It's possible. The handling the data is becomes a challenge.
Yeah, great. So many possibilities. It's fantastic. So I see another question. I think that's the last one because we are over time. So, but there is one question to go. So, is playing with deuterium less impacting the kinetics than adding bystander ions such as iodine to the solutions for calcite precipitation?
So first, deuterium is water, and the dissolution of the kinetics of dissolution and precipitation in heavy water is not very different from water. The only advantage is that the density difference between heavy water and water, so here we use either heavy water or a mixture of the two, the density difference is less than 5%. Whereas when you use the iodine, usually you need to use quite a high concentration. So if you want to do fluid mixing with iodine, you have two fluids with a quite different density. Normal water and the water with 3 molar of iodine, it starts to be, the density difference is different. And then you can have the potential iodine interaction with the other ions, but iodine is quite inert, so I don't think it's a major problem. Here we use deuterium water really because of its very specific property with neutron. It's possible to see it compared to normal water.
Yeah, you were looking for the difference that it could make kind of like a resolution thing. See the difference between the two fluids. Yeah.
So what you see is mixing of water with water here. That's what we see.
Indeed, which should be similar.
Yeah.
Okay, thank you. So just one thing, as I said, this was the last question, but there's one thing that one person asked, just to clarify, did you say millions of 2D radios per second?
Yeah.
Yes, okay.
It's an ultra-fast camera, and it's filming an explosion at, between so 2 and I think the next one will be 20 million images per second. So it's a very short experiment. The entire experiment lasts 10 to 100 microseconds.
Wow. Okay. Fantastic. That is ultra fast. Yeah. Great. So with that, I'd like to thank our speaker of today, François Renard. Thank you very much for a very nice and very clear talk. And thank you all for joining us today with this Excite Academy webinar. And we hope to see you next time. Have a great day. Thank you.
Bye-bye. Thank you.